Okay, good morning, Y10 Trip scientists. Welcome back to the Plants 2 topic. Today we are looking at the question, what is the effect of light and gravity on a seedling? This is part of the required practical activity, and unfortunately we are not going to be in the lab to be able to do it. I'm going to run through in as much detail as I think you're going to need. However, you will have the opportunity to do this when we do get back into the lab, as it is much easier to remember if you've done it yourself. Okay, so the driving question is, what is the effect of light and gravity on a seedling? Can you please put this driving question at the top of your page, make sure it's underlined to help keep your notes organized. Keywords for today are hormone, auxin, tropism, diffuse, phototropism, germination. All of these words we will have talked about last lesson, so if you're not sure on those, perhaps a bit of a recap on that would be useful. Variables, because this is an investigation, including your independent variable, dependent variable, and control variables. Okay, why are we learning about plants in the first place? Well, almost every ecosystem and animal, including humans, depends on plants for energy. By understanding how plants work, we can improve the yield of crops and help protect our environment. Brilliant. Plants are very important. Right, your starter, to get your brains going, I would like you to pause the video and have a go at defining the word tropism, gravitropism, phototropism, and germination. You do not need to write these down, just to get a quick definition in your heads before I show you my definitions for these. Okay, hopefully you've paused the video, had a go. Let's have a look at what my definitions are. Well, tropism is the turning of an organism in response to a stimuli. So a stimuli is kind of what the environment is like, which will then be affecting the organism. In this case, tropisms really affect plants. So gravitropism is the turning of an organism, changing direction in response to gravity. So positive gravitropism is growing downwards towards the center of the earth, and negative would be going upwards away from the center of the earth, away from the pull of gravity. Phototropism, and this is going to be the main focus of today's lesson, is in response to light. So turning towards the light would be positive phototropism, and negative phototropism is moving away from the light. And finally, germination. Not a word that we've necessarily come across. I wanted to test you, see some of you will have known this. And it's kind of the beginning of plant growth out of a seed. So to germinate a seed would be to leave it in kind of a warm area, usually wet, which is where you would see the first roots coming out of the seed and the first shoots coming out the top. Depending on the conditions after germination will depend on how well the plant grows and if it grows anymore. So here's some photographs of germination. The one on the left is underground. This is what hopefully would naturally happen in gardens, etc. You've got a seed, germination happens just after the seed, and you end up getting a seedling, which is kind of the first growth, but you've definitely got roots, you've definitely got leaves growing. In the case of the required practical, we're not going to be doing it outside in the mud. In fact, instead of using soil as the medium we're growing it on, we can just use cotton wool. So using something like mustard seeds, we can actually or perhaps cress, we can germinate seeds on cotton wool and actually have the first growth. The seedlings can grow on that. I think this is passion fruit seeds, in fact. Right, bit of a recap again from last lesson. Phototropism, what does that mean? And it is the response to light. In the freaky broccoli picture up here, it's growing towards the light. It isn't bending, as we learned last time. It is just growing towards that direction. Okay, so we go positive phototropism, growing towards the light, and it's the tip of the stem which is where the hormone is produced, which then controls the direction of growth. The reason for this is to maximize the amount of light available for photosynthesis. And we are going to go through photosynthesis again today, although it is something for the first plant topic. I mentioned hormones a second ago. In last lesson, we learned about auxins. Auxins are one of the hormones used by plants for growth. It's made at the tips of the stems and the roots. So the very tips of the plant, and this is actually where the growth comes from, the meristems. In the shoots, any concentration promotes growth, and in the roots, high concentration inhibits growth, and a small concentration promotes growth. I just want us to focus on the shoots for the time being, as this is what we're going to be investigating. So again, increases growth in the shoots. Right. Again, the mechanism for this is that when light hits the tips of the shoot, sunlight causes the auxins to diffuse away. This means that the shoot tip in direct sunlight has the lowest auxin concentration, and the area that's in the shade, the area that doesn't have light directly on it, has the highest auxin concentration. Because we know that auxin increases growth, then the shaded side is going to grow more than the side in the sun, and therefore there will be lopsided growth. The side in the shade will have elongated cells, that means longer cells, which will kind of push the direction of growth towards the light. So again, this is not bending, but rather it growing towards the light. Right. For the required practical, taken straight out from AQA, they said this: you need to be able to record the results of the seed's length measurements and create a labeled biological drawing to illustrate the effects of light or gravity. Our investigation has the question: investigate the effect of light on the growth of newly germinated seedlings. So, as with every single investigation, we have got our variables. Now, hopefully from Key Stage 3, we can remember that the independent variable is the thing that we are changing as scientists. And the dependent variable is the thing that we're measuring. An investigation really would be, how does one thing affect something else? So your independent variable is the thing that we are changing, and the dependent variable is the thing that we are looking at the change in. Importantly, we must have control variables, and these are things that we must keep the same, as they might also affect the dependent variable, but they are not the focus of the investigation. The focus is the independent variable. Okay, so in this case, in our investigation, investigating the effect of light on the growth of newly germinated seedlings, the independent variable is then the light level, how bright the light is, or where the plant is, depending on how much light it's getting. The dependent variable then is the thing we're measuring. That would be the height of the seedlings, or the height, or how much growth the seedlings have done, measured in centimeters or millimeters. The control variables are all the things that we must keep the same, all the things that might affect the dependent variable, all the things that might affect the height of the seedlings. And so these are the other things, other than the light level. And that could be the temperature. It could also be the number of seeds that we put into the growth medium. It could be the amount of water. Any other things that will affect the height of the seedlings that are not light level. Okay. Practically, you're going to have to do this. So how are we going to be able to change the independent variable to be able to then make this comparison? Well, we can easily change the light level by changing where we grow the seedlings. So, for instance, if we put it in the cupboard, it would be completely dark. If we put it on the windowsill, that's spelt wrong. If it's put in the windowsill, then it will obviously get the most light, and hopefully we'll be able to grow the most. And we would also want one that has partial light. Now that could just be with a filter over it, but probably the easiest thing to do would just to be putting it in the light for some days, putting it out of the light for other days, or perhaps put it in the light for some of the time, out of the light some of the time. How can we practically measure the dependent variable? Well, the height of the seedlings we can measure using a ruler, simply. And to control variables, those things would be the room temperature, the number of seeds, and measuring the volume of water each time. So again, dependent variable measured in centimeters or millimeters. The control variable, keeping the room temperature, that's measured in degrees C. You could just change the thermostat, and the volume could be measured that you water each batch in milliliters. Okay, what I'd like you to do now is to pause the video and just have a go at matching the variable to the definition of what the variable is. When you come back, we'll go through the answers. Okay, the independent variable is what we change, the dependent variable is what we measure, and the control variable is what we keep the same. Okay, so our investigation, this is the method. You are going to need to know the method by heart because you basically, it's required practical, you're going to need to know it inside out. And this is what we would be doing. So as we put cotton wool into 3 petri dishes and add the same volume of water to each dish. Then add 10 seeds to each dish and place them in a warm place where they won't be disturbed. So this will allow them to germinate. Allow the seeds to germinate and add more water if the cotton wool dries out. So first step is just germinate some seeds in 3 different petri dishes because we are then going to be changing the light conditions for each of those, and that's our investigation. Once the seeds have germinated, ensure that the petri dishes each contain the same number of seeds and remove any extra seeds if necessary. So for instance, there might only be 8 in one of them, and that means that you make sure that there are only 8 seeds in all of those. One petri dish will sit in full light on a windowsill, the second will be in a dark cupboard, and the final dish will be placed in partial light. Now, depending on what resources you have will depend on how you can do the partial light. But yeah, for now you just need to know that you've got one in the bright light, one in the pitch black, and one in partial. So there we are, there's the box, we put that one in. Second seedling in bright light, and the second one is only in partial light. Every day for 1 week, measure the height of each seedling and record the results in a table. You must record the height of the individual seedlings on each day. So literally going around and measuring the height that the seedlings grow. Finally, calculate the mean of the seedlings each day and compare the mean heights of the 3 different locations. And then plot the graphs against height, against time, and you may compare the growth of the three batches of seedlings. Okay, as with all investigations, we are going to have to record the data as we go. This would be an example of the table that we would make to grow it. Down the side, you've got the days, day 1, day 2, day 3, all the way down to day 7. Why? Because it's a week. And then I've got 1 to 8. This would be if there are 8 seedlings, and there will be space underneath to write how many millimeters of growth it's got. There's a space down the side for the mean, and we will be calculating means next lesson. But for now, I just need you to memorize the type of table we are going to use. So again, the 4th point of the method: every day for 1 week, measure the height of each seedling and record the results in a table. You must record the height of the individual seedlings for each day. So this is why it looks like this. Now, of course, this is only one of the tables for full sunlight. There will have to be 3 tables in total: 1 for light, 1 for no light, and 1 for partial light. Calculate means you add up all of the heights of all of the seeds, and then divide them by how many there are. So, for instance, divide by 8, and then that will give you the mean. That's an average. Right. See what you can remember. What could the control variables be for this investigation? Please pause the video, have a read of these boxes, and pick one that you think is the most suitable. Okay, welcome back. Well, light level is the independent variable. It is the thing we're changing on purpose. Control variables are the thing that we must keep the same, so it isn't this one. It's not light level, it's not light level, it's not light level. It is going to be B. The amount of water that you put in might affect the growth. The number of seedlings might affect the growth, and how spread out the seeds are might affect the growth, as well as the temperature of the room. As we know, temperature affects photosynthesis. Right. Why must we keep control variables constant? Why do we need control variables in the first place? Why do we have to care about anything other than the independent variable? Well, this is because we're trying to deduce the effect that the independent variable has on the dependent variable. Simply, how does light affect growth? Control variables must be controlled because if they're not, they might influence the dependent variable too, even though they are not the focus of the investigation. So this is validity. This means a valid test only has the independent variable affecting the dependent variable. It would be unfair to have one of the batches of seedlings in the dark, but also in the warm, compared to the seedlings on the windowsill, which might be in the cold, but also in the light, because the temperatures are then different between each seeds, stops the test being fair, and then it would be invalid. So, why do we control these variables? Now, I've said that they all affect the growth. They all affect the dependent variable, but actually, I'd like you to have a quick think. How would the amount of water, the number of seedlings, how spread out the seeds are, and the temperature literally affect the growth? What's the explanation for that? Pause the video, have a little think. You don't have to write anything down for the time being, and I'll show you when you get back. Hello, welcome back. Right, the amount of water will affect the amount of photosynthesis the plant can do. Photosynthesis requires water as a reactant. The number of seedlings the seedlings may compete for light or for space or for nutrients, and therefore you must keep the number the same. Again, because if the seedlings are really spread out compared to ones that are right next to each other, because seedlings might compete for light or space or nutrients, that again, how spread out they are may be a fact that affects the dependent variable. And finally, the temperature, of course, will affect growth because temperature will affect the rate of chemical reactions. Photosynthesis is a chemical reaction. The closer to the optimum temperature, the faster the rate of photosynthesis and therefore the greater the growth. So temperature must be kept the same too. Right. This is the part of the lesson where I'd ask you to speak to the person next to you, chat to your partner, and try to explain something out loud. Fortunately, we are not in the lab, as I said before, as you know. So I'd like you to go and find somebody and tell them. This could be, a parent, brother, sister. It could be your pet. It could just be a neighbour. It doesn't really matter who. But I would like you to actually tell them the answer to this question. What are the variables for our investigation? Our investigation is to investigate the effect of light on the growth of newly germinated seeds. What is the dependent, independent, and control variable, please? Pause the video, go tell somebody, and then come back and I'll show you what those are. Okay, hopefully you've just had some quite positive conversations about this. By talking, by trying to teach somebody something or explain something out loud, you are consolidating that information, and it really is going to get stuck into your long-term memory. This is the reason that we do this. So it's very important that you do actually go and have these conversations, otherwise it's a waste of time, really. What are the variables for this investigation? Well, the independent variable for this investigation are to place the plants in light or dark or partially light conditions. The dependent variable is the mean height of seedlings, and the control variables are the number of seeds on each dish, how much they're spread out, and the volume of water the seeds are given. The temperature is also important. Right, it is now time to do the writing phase. This is the part of the lesson where underneath the driving question, underneath the date, you have a go at answering the objectives one at a time. So, your describe question is to state the dependent, independent, and control variables for this investigation. The explain is design a results table to be able to accurately record your results and prepare an axis for a graph. You do not need to actually take the data. I'm not asking you to collect anything, but can you draw the table to, if you were going to? Can you have a go at writing the full method of the investigation using your understanding of the variables? Now, it can obviously vary from the one that I've shown you. However, a good method would be if you were to follow those instructions, you would get the results that you require. And then finally, have a go at explaining the necessity of a control variable and what effect each control variable would have on the growth of the plants. Give yourself between 10 and 15 minutes on this. Start with the describe and work your way down. Good luck, year 10. Pause the video. We'll go through answers when we get back. Hello, welcome back. This is the part of the lesson where we are going to go through our answers. Give yourself ticks if you've got them right. And if you're missing any information, make sure that you get that in with your different coloured pen. Right, independent variable. This is where the plants go. So in the light, in the dark, or partially light conditions. The dependent variable is the mean height of the seeds. If you haven't got mean height, that means the average height. Because we're using lots of seedlings, we're not just adding them all up together, we are taking the average, we're taking the mean height of seedlings. The control variables are the number of seeds in each dish, how much they're spread out, the volume of water given to the seeds, and the temperature that they are kept at. All of these things must be the same. Right, the table. Well, we mentioned before that every day for a week you measure the height of each seedling and record the results in a table. You must record the height of the individual seedlings on each day. This table would allow you to do that. If your table does not have 7 days, if it does not have space for all of the seedlings, I've got space for 8, but you could have more, and a space for mean, and the title for these columns, which is the height of the seedlings in full sunlight, and the title for this column being day, and you need those. You don't need to do 3 tables. However, if you were to do this investigation, you would have to have one for sunlight, one for the dark, and one for partial light. A note saying 3 tables must be made, one for each light level, is necessary if you haven't done 3 tables. Okay, there we are. Right, this is the method for investigation. Number 1, put the cotton wool into 3 petri dishes and add the same volume of water to each dish. Add 10 seeds to each dish and place them in a warm place where they won't be disturbed. Doesn't matter if you've said 10 or however many, but as long as it's the fact that I've said 10 means that they'll all be the same. To allow this, then allow the seeds to germinate and add more water if the cotton wool dries out. 4. Once the seeds have germinated, ensure the petri dishes each contain the same number of seeds and remove any extra seeds if necessary. 1 petri dish will sit in full light on a windowsill, the second will be in a dark cupboard, and the final will be placed in a partial light. Every day for a week, measure the height of each seedling and record the results in a table. You must record the height of the individual seedlings each day. Then, calculate the mean of the seedlings each day and compare the mean heights of the 3 different locations, perhaps putting it into a graph. If you're missing any of these points, or if you'd like to copy it out now, pause the video, and this is the method. It's easier to remember if you've done it before, and you haven't, okay? Just bear that in mind. Not easy to memorize. And then finally, what's the point of a control variable, and what are they, and why do we have them? Control variables must be controlled, because if they're not, they might influence the dependent variable too. Even though they are not the focus of the investigation, this keeps the test valid. That should really be a key word for today. The amount of water is a control variable. This is because the amount of the water will affect photosynthesis. Plants need it for photosynthesis. In the same way, temperature will affect photosynthesis as it affects chemical reactions. The closer to the optimum temperature, the faster the rate, and therefore all temperatures must be the same. And the number of seedlings and how spread the seedlings are will affect the growth, as the seedlings will compete for light, space, and nutrients. So there we are. That's your combined section. As always, I'd like you to write your today I achieved sentence. So that would be today I've achieved, and then write describe, explain, use, or create in this section, and then say because you could. And depending on which one you've chosen, you need to copy in literally what it is that you've done. For instance, today I've achieved explain because I could design a results table to be able to accurately record your results and prepare an axis for a graph. Okay, This is not easy, a ton. Well done for your patience. We are going to do more on this required practical next lesson, and hopefully that will help us stick into our brain. As always, you've got a 10-question Show My Homework quiz. Ensure that you do that, please. I'll put some resources on Show My Homework as well to give you a hand with those. Okay, good luck with the quiz. I'll see you next time for more work on the required practical. Thank you, and bye-bye.